Welcome to the Healthy Enterprise. My guest today is Ed Fleischer. Ed is a quality control executive who has been working in the biotechnology environment for many years. He's now taken his experience to start his own consultancy called Beachside Compliance Solutions, where he specializes in analytical and quality control issues. Ed is also a very good storyteller, and there's lots to take in. So let's get started. How you doing, Ed? Thank you for joining me today on this episode.
Well, Heath, thanks for having me on. I'm Ed Fleischer. I have a one-man consultancy called Beachside Compliance Solutions. Beachside because I live 2 miles from the beach. I'm down in beautiful, sunny Southern California. I live near the beach, but I never go there, which is, a shame. But
what?
That's the way it is.
What's your closest beach? Huntington?
I live in Carlsbad, so
okay.
There's a fabulous beach, and if you ever get down to Carlsbad, everyone, it's a beautiful place to visit. Legoland is here, so we've got some attractions there, big flower fields, beautiful beaches.
We almost went to Legoland. We were down in California a few years ago with my wife and our 2 sons, and we hit all the hot spots. And Legoland, we kind of burnt out. We'd just finished. We were on our way back from San Diego, and it was towards the end of the trip, and I literally couldn't convince everyone to go to one more amusement park. So
yeah, I could see that. But yeah, we got SeaWorld. There's a zoo. We've got the safari park.
Wow.
Some great museums, some beautiful parks, and beautiful ocean and beaches here. So,
Ed, you got to get to the beach.
Well, like I said, I get in my name, and we went when we first moved down. We went quite a bit, but lately everyone's been so busy, it just isn't happening.
Okay, well, tell us what you're busy doing. You've got a long history in.
Yeah, so I'm an old time analytical guy. So when I got out of grad school in 1983. I got a job as a research associate in a small company that was working on interesting new technologies to make vaccines, subunit vaccines. So instead of the old killed virus, we were going to go ahead and clone the bits that would make a good vaccine and put them together. Unfortunately, the company ran out of money. And then I went to another vaccine company, and then I was recruited by an old boss of mine who. The first company I worked with, he had just joined a company at the time called SmithKline and French, famous for making Tagamet. They were building a new biotech division, and he wanted to hire someone who could work with proteins. And I had worked for them before, so he recruited me there. And I stayed there for 17 years. I worked for SmithKline and French, and then SmithKline Beecham, and GlaxoSmithKline, 3 different companies, and I never left the building.
Wow.
I was in building 30 at Upper Marion in Pennsylvania the whole time. And worked on all kinds of stuff. Analytical method development, we validated methods. We supported IND, so investigational new drug applications, mainly on the protein side. I did do some small molecule work, but most of it was on the large molecule side. And once the Glaxo merger happened, they got really serious, and they basically split their biotech group away from a general development group. And I went with them, and I ended up running a QC group that was making all kinds of investigational products for the Glaxo portfolio. I actually wrote the proof of structure document for a drug called mepolizumab, which is on sale still today.
Is that right?
So I got to work on that. It's a asthma drug for eosinophilic-driven asthma and stuff. When we worked on it, we couldn't get it to work in the clinic. We called it the world's most expensive placebo because it was safe, very great, but we couldn't get it to work. And then my understanding was a clinician in Canada saw the data, and he said, "I know what you're doing wrong. You're testing it with everyone with asthma, and your drug works on a specific type of asthma." And they redid the clinical trial, and lo and behold, it worked. And it's still on the market, going strong. It's a great product.
Amazing.
So I was on the East Coast all that time. Then I got recruited to go out to Amgen out west. You know how these things happen. I was at a meeting. It was an FDA workshop, and I ran into a guy I used to work with. He had left Glaxo during the merger, I think. So I hadn't seen him for 3 or 4 years. I ran into, we had lunch. He goes, "Oh, hey, I might have something for you. give me your contact number. So I gave him my card and totally forgot about it, right? Said,
Of
oh,
course,
this
yeah,
happens
yeah,
all the
as you
time,
do.
but there's never any follow through.
Yeah.
So like a month later, I get a call and it's a headhunter, recruiter from Amgen saying, hey, I got your number and we'd love to talk to you. You know, could you send me a CV? So I sent him a CV, and then they did a couple phone interviews, and then they flew me out. And this was for up at the Amgen facility that was up in Seattle. I toured Seattle the 2 days it didn't rain. So it was
Wow.
beautiful. It was beautiful. I was up there in September. I think it was September, end of September, beginning of October. Beautiful day there. Really liked it. They made a really nice offer. I had to convince the family this would be a good move. So we went out and I took my wife and my two daughters out hunting, house hunting, and it rained all 4 days. It rained so hard, the rain was coming up from the ground.
Oh my gosh.
It was a Pineapple Express. I don't think they call them that anymore, but I
They
never
no, they're
experienced
called
that.
atmospheric rivers
Yeah,
now.
atmospheric rivers now, and it just rained the whole time. Luckily, we found a house we all liked, and then we moved out there, and I was there for about 4.5 years. Amgen decided to close the plant. It was a GMP manufacturing plant and move everything out. So I was kind of a free agent. I interviewed a bunch of places. I got recruited by Genentech to come down and run their QC group in Oceanside, and that's where I still live. It's down in Carlsbad, which is just south of Oceanside.
Hmm.
Genentech is a beautiful biologics manufacturing plant in Oceanside, and they've upgraded it. They've added some new single use technology. It's a beautiful facility, one of the nicest ones I've ever seen and worked at. So I was there for a while. Then I went to a corporate role there, and I spent a lot of time up in South San Francisco. And then I got recruited to go back up to Seattle to join a company at the time called Cgen. They're now owned by Pfizer. And when I asked the family, hey, who wants to go back to Seattle? Surprisingly, everyone said, well, you can go. We'd prefer to stay in sunny California. So I did a long distance commute for 3 years.
Wow.
I had an apartment up there, and I would fly up and spend as much time as I could to do face to face stuff, and then I'd come back home and spend some time with the family here. And then COVID hit,
Yeah.
right? And we had
That
to
sent you back
refigure
to
out
California.
how do we do this remotely. So I spent a lot more time doing remote work there. The success story there was I was hired to get the group ready to deliver. We had three products that were going through the approval stage, so three BLAs that were going out to get these done in 3 years, and all three of them were approved while I was there. I like to say I had a hand in some of it, but that was very successful. So this was a company that specialized in antibody drug conjugates,
Hmm.
which I think everyone's hearing about now. It's a real revolutionary treatment. It's sort of that silver bullet technology where there's opportunities to treat oncology in a very different way, maybe certainly with much less chemotherapy, and very targeted. So I was there for 3 years. We launched all three, um,
Is that more
and then I
custom?
got a
Is
and by then the commute was kind of grindy.
Yeah.
So I moved to Gilead, who happens to have a site right across the street from the old Genentech site I worked with. They had an ADC, an antibody drug conjugate, and they were launching. They wanted to beef that up, so I was there for about a year and a half, and then they announced they were going to close the Oceanside site down. And I said, "Well, I'm not moving. I'm done." And I said, "Well, I'll open up a consultancy and see what happens." So. The good thing is, I knew a ton of people in the business. I've been all over. I went to almost all the Amgen sites. I've been to all the Roche sites. In LinkedIn, I had literally thousands of contacts. So I reached out to some people, and there's big consulting firms that hire what were called 1099 employees. Not
Yep.
derisively,
Yep.
but they hire people with my skill set. So I did a big job, a couple months work with one of the consultancies, and then I had a bunch of travel I wanted to do, so I didn't work for a couple of months, and then I got another call from another consultant group, and they needed someone to come in and visit a site and spend a couple weeks assessing their validation program. So I flew in, met some wonderful people, read a bunch of documents, talked to some people, made a bunch of recommendations, and And then I got another consultancy called, and then more and more. And like I said, I've been just busy the whole time, which is great. It's kind of interesting. It's not what I thought it was
Right.
going to be. It's was different in some ways. And the thing I thought was, oh, you fly in and you do some work and then you leave. And that's what we used to say. We hired consultants. They don't care what the outcome is. Well, it turns out they do care. I really do care about
Hmm.
the outcomes of the places I work, and I follow them. And I stay in touch with people to see, hey, we proposed these fixes or we did this work. How did it work out? And one, I went, like I said, I flew in. They wanted me to evaluate their validation program. I spent a week on site, and then I spent about a week writing a report. Giving them some feedback, and then they asked me to continue on, kind of ad hoc. Hey, an analytical problem came up. Could we book some time with you? And so that's been really successful to do that. And I think the key things is you need to stay current. Regulations are always changing, guidances are coming out, and you need to be able to really understand what the customer wants and then deliver to what they need. They don't want more, they don't want less, they just want, kind of what they want. And sometimes they don't know what they want. You have to help guide them there. So it's been really crazy. Like I said, I'm doing one right now. It's a big job. They hired literally dozens of consultants to do some of these assessments and stuff, and it's been really fun. You get to see, these are companies some of them I've worked with before, some I haven't. It's no surprise I run across people I've worked with before in my prior life and stuff. So it's been really fun and exciting. Um, the downside is if you don't work, you don't get paid.
That's right.
So it was a shocker to me that last Labor Day was the first time in 41 years I did not get paid on Labor Day to not work on Labor Day. It was stunning to realize that, hey, if you work at a company, they pay you not to work some days.
That's right.
But it's worked out good. So there's a lot of flexibility. You can turn down jobs if you don't want to do it.
Yeah.
You can pick and choose somewhat, but it's been really fun and really exciting to do it. I wasn't ready to retire. I still thought I had some juice left.
Sure.
And it's worked out pretty well. Like I said, I've met some people that I worked with. One place I went, I had lunch with one of my old bosses who was at the other company. It was great to see her. So it's really good to stay in touch. And, the business changes. I mean, the stuff I used to do even 10 years ago is somewhat outmoded now. So you really need to, stay current
Yeah, you
with
got to
what's
stay
going
current.
on.
And how do you stay current? What do you use? What do you use to keep up
Well,
on
there's all
things?
kinds of websites and stuff. So, you can subscribe to sites. I'm very active. Well, I was more active than LinkedIn when I had more time, but LinkedIn is always a wealth of things. You can subscribe to different like aggregators. So like Fierce Biotech, Pharmaceutical, they aggregate stuff up and send stuff out. I still pop in on the FDA website every couple weeks just to see what they're posting. There's all kinds of, I was in the GMP space for so long, the regulated part of the business that there's various things like the pink sheets that come out and tell you what's going on in the regulated world. I think everyone that's going to tune in here knows that ICH came out with new guidance on stability that just got released.
Hmm.
It's the first, I think, in I want to say 15 years. They updated that. I could be wrong with the dates, but it's been a while, and they consolidated a whole bunch of guidances that were spread out amongst different sections of the ICH world, and they consolidated one into one big document. So I'm just getting ready to download that and read through the changes and see what's new. When clients call, I'm sure they're going to have questions about that. How do you
Sure.
interpret this new guidance, and what could you recommend?
So, what kind of innovations are you seeing now in this space that you didn't see, 5 years ago? Like, what kind
Well,
of things
certainly
are shaping the
AI is
industry?
certainly popping up.
Yeah, but
So,
yeah,
to no surprise, when you type anything now, it's filling in your sentence as you go.
yeah,
And sometimes it's not what I want to write, but they're suggesting things. It was really interesting. We were at a offsite gosh, it was about a year and a half ago, and we were kicking around like what a global strategy would look like, and somebody opened up Chat GPT and typed in global QC strategy, and it spit out something that was probably 95% good, which is disconcerting to me, somewhat disconcerting to me because, you know, that's my expertise.
Right.
And you could replace it. Of course, there were errors and stuff, and you'd have to customize it. But
Sure.
if you were trying to get a lot of information gathered quickly, assuming the source is, the source subject is pretty good, and QC's been around a long time, you could probably generate some pretty good information without a lot of work.
Well, I mean, you're like you said, the intelligence is not surprising in the sense that I've had many of these conversations over the last few weeks. Some of the comments that are coming back are that if we look at it like this technology is really something that was built by everybody.
Yep.
It's a collective. It's all our collective knowledge as a species all piled together into one place, which if you researched it for your entire life, you'd never scratch the surface of it. But collectively, we're dumping all this information into a
Yeah.
database that is taking everyone's thoughts, considerations, research, strategies, outcomes. Whatever information is out there is being pooled, and so when I think about that, it's like really, it's quite an incredible um, possibility, an opportunity that
Yeah.
we have all that information at our fingertips, where we can now type things into chat gpt or whatever program. There's so many of them now. We always revert to that one because it was one of the first
Yeah.
ones to come
That's
out.
the one that I'm most familiar with.
So they and you get back a collective perspective. And it's I mean, I don't remember actually who said that, but I'm actually really glad they said that because it's kind of shifted my outlook on AI and where that's going. And I think it is going to be the future. It's our collective intelligence, really.
Well, and some of it is, we deal in data in the pharmaceutical world, the biotech world. It's data driven,
Right.
and we've got a lot of data that sits there that there's probably some value. We haven't figured out how to harvest that value. One thing that we do in QC, we run the same tests over and over and over, either on the same molecule or it's slightly different on a different product. And what do you get when you run something, literally hundreds of thousands of times in industry? If you're making a monoclonal antibody and you follow the guidance, everyone is going to look for, aggregates. So they're going to use size exclusion chromatography to do that. So everybody who's developing a biopharm product is going to run some version of a size exclusion chromatography method on this, which means if there's literally thousands of monoclonals in development, there's probably hundreds of thousands of data points that could be drawn on and help you understand, hey, if you see this, this is what it means to be a well characterized molecule. You'll know this aggregation or this dimer or this appearance of some other form in your product. And there should be no surprises there. That's what I've done for 40 years. I run these things. Now someone who has very little knowledge could type this in and probably develop a very robust specification
Hmm.
that would enable them, to test and release the product, where we used to have to run these things over and over and over again to build up the data. And that's one thing that all these companies that do this, they've got literally millions of data points sitting somewhere. And if it's in QC, it's in a LIMS system, laboratory information management system, with a lot of other information that could be useful. And we're just starting to harvest that stuff. My question is, if I run the same test all the time and I get the same answer all the time, what is the value of me running it all the time?
Right.
It costs money to do.
Hmm.
Is there other ways that we could say, if the process runs in its design space and the raw materials have been analyzed and tested, and they work the way they're supposed to, what's the likelihood of me finding anything that's wrong? It's
Right.
very slim, but it costs to do these tests. It takes
Hmm.
time. It takes manpower. You've got capital equipment involved. Are there opportunities to possibly not do so much testing? And certainly, the agencies are willing to look at that. Our the I still remember when it was in this concept of design space. The quality design space was if your raw materials worked at a certain space and your manufacturing delivered in a space, as long as the product was in that space, it couldn't be wrong. Why would you have to test it? All you'd have to do is prove its identity and did it work? All the other stuff wouldn't have to be done. Well, we're not
Right.
there yet.
Right.
And that might be where we're heading
That might be
because
where
now
we're
there's
going.
another way to unleash all this data to do that.
So over your years of work, what area do you like the most? What's your sweet spot?
Oh, there's a lot. There's been some one thing is you get to work with some truly extraordinary people.
Yeah.
I mean, uh, I was never the smartest person in the room. There are some really smart people out there that really understand all kinds of things. You get to work with really cool technology.
Hmm.
And it's always changing, right? You get to work with, if you're in the commercial space, you can be working with, 25,000 liter bioreactors, just huge magnitudes of volume of water and product. And you get to work on stuff that really impacts people's lives. I always have
Right.
a story that really got to me. I happened to be in London, getting ready to go up to Cambridge. There was an analytical symposium held every year at SmithKline Beecham, and they would pick people to come up and give a talk about what they're doing, or mix and mingle. And I was selected to come up. And we were told, you shouldn't wear company swag when you travel to London because the SmithKline at the time didn't have a great reputation with animal rights people. We did do animal testing, and it was not unusual for people to get, abused if you wore that. Well, I was an American and I didn't listen, so I had my SmithKline Beecham hat on, baseball hat. That's how you can pick Americans out. They're the only people that wear baseball hats
That's
in Europe.
right.
So I'm standing on this train platform, and this lady comes up and she goes, "Do you work for SmithKline Beecham?" And I said, "Yeah, I do." And she goes, "I want to thank you. You gave me my son's life back." And I go,
Wow.
"Really? How?" And he goes, "He was a severely depressed individual. He was involved in one of the clinical trials for Paxil, which was a drug to treat adult depression." And she said within 2 weeks, 3 weeks, he was his old self again. We were so worried that he was going to kill himself because he talked about it. And he took this drug, and he's my son again. I want to thank you. Very
Wow.
powerful.
Very powerful.
And this wasn't like we brought. Usually we would bring patients in, and they would talk about how they've used the drug and what it's
Sure.
done. This
Case
was
studies. Yeah,
this was
for sure.
5 minutes on a train platform in 1997, I think,
In
something like
London,
that.
England, with
Yep,
like
really powerful stuff.
for the two of you to cross roads at that exact moment, and for her to have some sort of association with the company you work for. I mean,
Yeah.
and you were told not to wear your logo on your hat, and had you not done that, she would never have known. I love that's a crazy story.
So
That's
it's very powerful though because
yeah, very powerful.
the medicines we deliver, they may not cure you, but they certainly give you hope.
Yeah.
We were racing to get an oncology drug approved, and the chief medical officer said, "People are counting on us. We cannot delay this. We have people that are literally in the clinics waiting for this to be approved, so they can take our medicine because they have the hope that this drug, it might not cure them, but it will certainly delay, their end game, and maybe they'll be alive to see their daughter or son get married, or
Hmm.
they'll see that grandchild take their first step, or they'll do something that's important to them, and we can give patients hope by delivering these products, and that's a real powerful motivator, right? To
Hmm.
to, to say that there are people that are depending on us to deliver these medicines. and we need to make sure they're safe and effective, and they can get to them in time.
Well, yeah, and I guess there's a bit of a misconception sometimes about big pharma, and they're the need is there. I mean, disease is quite abundant, and these are solutions, right? Maybe, like you say, they're not cures, they're not going to resolve the problem, but like you said, improve quality of life
Yep.
potentially for people. Certainly, in that young gentleman's case, you changed his life actually, and he had a lot of life ahead of him. So
Yeah.
yeah, it's you know, to get up and go to work every day knowing that what you're doing is actually going to have some sort of um, positive outcome for someone's life. I mean, that's why most people enter the healthcare, health tech,
Yeah.
medical system in the first place, is
Yeah.
you want to
Well,
make it. You want to
there
make a
was
difference.
the company that I was at previously. We were in a. I was they were in New Jersey. The team I worked with, and I would fly to Jersey every month, and all the conference rooms had people's names on them. And I never really thought about it. And then one of the longtime guys there said, "Do you know why that is?" And I was like, "No, I just people liked them." He said, "These were the first 12 people to take our medicines in the clinic. And most of them are still alive. And, they were diagnosed with breast cancer,
Wow.
gosh, 4 or 5 years ago,
Wow.
and most of them were still alive and taking the drug. Either treat them and put in remission, or they were
Right.
still able to. And I thought, wow, that's really something. And then I was there, and they got the news that one of the first people to be dosed, and this would have been like in 2017, 2018, she just passed away.
Hmm.
Um, which was sad. But I thought that was really cool that they memorialized, the first 12 people to
Yeah.
take the medicine and gave them names. And I said, why isn't that more well known? How come I didn't know this? I'd been visiting the site for 6 months. I
Yeah.
saw the names on these rooms, and I didn't ask, why are they named that?
Right.
And there was no story around it. So it was really nice that he shared that
Yeah. Yeah.
stuff.
That is important. So you're from your perspective. I mean, I mean, you're not a founder. You're not, in upper management. You're actually you're at the front line working on these medicines and at the at the molecular level.
Well, yeah, but
So,
wait. So that was a long time ago when I got into management.
right.
So I was, I used to run big groups. I was a senior director and stuff, and
Okay. Yeah.
I couldn't probably turn an HPLC on anymore. But it's important to understand the fundamentals of what we're doing and stuff.
Yeah.
That's why I don't know anybody who still doesn't keep up on a lot of the the real key basic applications of the work.
Right.
When I used to do work in the lab, the changes have been so tremendous on some of the analytical tools being used. New detectors, moving to these micro volumes instead
Hmm.
of large volumes for testing. Rapid answers. I mean, we used to do one of the tests that you have to do in the biological world is to look for adventitious agents
Hmm.
that may be transformed or transferred through the process. So if you're using live cells, they could be infected with viruses
Sure.
because they're live cells,
Yeah.
right? So one of the tests is looking for mycoplasma, which is a little tiny organism that may or may not be present. Luckily, we've never found it in any place I've worked, but if you get it, it shuts you down.
Oh wow.
But so the old test that everyone accepted was this broth culture test. You'd get your sample, and you put in this broth, and you mix it up, and then you grow indicator cells, and then you look at these cells like 15 days later and 28 days later and compare them to healthy cells. And if you have the virus, the cells will be different. They'll be dead, or they'll have different morphologies and stuff. It took 28 days. So the clever scientist said, wait a minute. That means if I have a drug product, a drug substance that I'm making, and I want to release it, I can't release it for 29 days because I have to get this result. And that's true. So, we targeted 32 days to release a batch. Well,
Right.
what if it was in high demand and you wanted to get this out faster? Well, you were kind of stuck. So, this really interesting technology, PCR, comes along, polymerase chain reaction, where you can amplify bits of DNA and run tests to show if something's there. Eventually, they started doing this in mycoplasma. You can now run this test in your lab. You don't have to ship it anywhere. You can run in your lab and get the data in days
Amazing.
and
And
stuff.
speed that process up
Yeah.
dramatically. Wow.
So there's all kinds of stuff coming up that
Yeah.
they're looking at rapid sterility testing. There's the new therapy. It's not new anymore. It's been out for 10 years. The CAR T uh, therapy. That's human. A patient comes in. They donate their own material, plasma, blood, whatever it is,
Right.
and that becomes part of the product.
Oh, wow.
And it's fast. You have this stuff in. You need to get this back to someone in 2 weeks. How do you do a 28-day test if the patient is going to get it back in 2 weeks?
Yeah.
So there's all these new technologies to speed up these rapid technologies to speed up the process, so these life-saving products can get back to patients.
Yeah. That's amazing.
Yeah,
Yeah.
so it's
That's a
really and it's
game
really cool
changer.
because I remember doing PCR when I did it the first time. I amplified dog DNA because we had a big dog, and I was not big. I had a Samoyed. They shed like crazy. The hair got in the mixture. Boom. I didn't
Wow.
understand the techniques that you needed
Right.
to do that. So we know better now how to do this, and they made some amazing things. You buy kits, pre-made kits and stuff. You put your sample in, boom. You get this data out. When I say quickly, you still have
Incredible.
to validate everything. It's probably
Sure.
going to take you a year to do it, but once you get it in, it can revolutionize how you do your release testing.
Yeah. That's amazing. Wow. Um, when you were leading teams back in the organizations you worked at, what would you say your leadership style was, and when you were running the show there?
Well, the feedback always was I was authentic. I am what I am. I don't try to change that. It doesn't work. It
No,
doesn't work.
no.
But I've worked with people that did. They weren't authentic. So very authentic. By then I had a lot of experience. I
Hmm.
had almost seen everything. Although every time I say that, I see something I never saw before. I like the technology. I like the aspect of building teams. I understand. I appreciate the wild diversity that the science world gets.
Hmm.
It was amazing when I'll go back to the Glaxo days. We had a team of 40 people. I was one of the team members, and we went. We were doing some ice breaking, and it turned out. So we're in Pennsylvania. Only two people on the team were from Pennsylvania, and there were 28 people from other states, and there were 13 or 12 or 13 from other countries
Wow.
and stuff.
Real
And
melting
we
pot.
had people with all kinds of wild experiences. People that did grad school out east, not just East United States, but out in Poland. People that worked in Japan, China, all over, and it was amazing to see how the bad word of DEI, the diversity part, uh, really came together. So it was great to have people with all kinds of points of views,
Hmm.
knowing they had you could make a safe space so people could argue and debate. They're not going to get nasty about it, but you could bring up diverse point of view. Hey, this will never work. I
Hmm.
remember we're having this thing. I proposed something, and the woman next to me said, "There's no way in hell that will work." And I turned to her and said, "You're going to eat those words because I know exactly what is because this experiment they couldn't get it to work." And I said, "I know you're doing wrong, and I will get it to work." And I did get it to work. But I didn't take it I took it as a challenge when she said that. I wasn't
Right.
insulted.
Right.
She didn't
Right.
think I was an idiot. She just didn't think it was going to work, but
Right.
it did.
That's an interesting thing about science is that it really is. It crosses all boundaries, doesn't it?
Yeah,
There are people researching things, some of the same things, all in every country in the world, and then sharing that information is where science actually has breakthroughs. Is
Yeah,
where,
because we all have the same, if you're working in the biological world,
yeah,
there's a lot of the same challenges. The regulations are set to address certain things. And then you have to meet those regulations. And there's always a reason
sure,
why. When people, when people badmouth the FDA, I said, hey, look, they have a job to do. And they do it well. I've never had a inspector I thought was unfair. I've been through 40 something FDA, EMA, we had Koreans, Chinese, Japanese, from all over. They come in and they have a reason to come in and look because there's bad actors out there.
yeah,
I remember I was at a workshop around raw materials, and this was when I want to say 2007, and some clever people in China figured out you could substitute something, and it looked just like heparin until you took it. And they had, literally, I can't remember how many people died in the States because they got uh, this counterfeit heparin product and
wow,
stuff.
wow,
And that changed how we did testing. They immediately said, "Hey, there's just some new things you have to do now to be able to release this stuff," because we found people that could find a workaround and
hmm,
stuff. So they've
interesting,
got a role to play. But yeah,
yeah,
one of the greatest things is you get to work with some really interesting people. I've got to go to some really interesting places. I mean, the company sent me to Singapore to give a talk. I used to go to Europe, oh,
yep,
gosh, 4 or 5 times a year, all over the states. You get to meet, like I said, you get to meet some amazing people, some really smart people, some people that know stuff that I didn't even know you needed to know.
So you were doing presentations, like speaking and doing
Yeah,
delivering
yeah,
talks. Yeah. Did
yeah.
that was a became a large part of what you did?
Well, it's something that if you're going to lead people, you need to get up in front and talk to them
Hmm.
and stuff. In fact, it was really interesting. This goes way back. So when I was in high school, my goal was to become a doctor. I wanted to be a doctor, and I was talking to this counselor, and he said, you should probably do something around public speaking. I said, like debate? Because I had no interest in debating. He goes, no, you should take an acting class.
Oh,
Because if you
really?
can get up in front and say other people's words and act, you'll be very comfortable talking to large groups. So I did. I actually took a year of acting
No kidding.
and stuff. And it was really useful
Yeah.
because you're right. If you can get up and say other people's stupid words, you can say your own stupid words. And that was one of the things that helped me move into management. I never had issues with getting up and talking about my work or presenting you know our team's work, being able to put together slide decks. You always have to talk to slide decks, and also making engaging talks. In fact, I got invited to go to England for this analytical symposium. The first year I was invited. I gave a poster. So we did some experiments. We said, hey, this is what we were set up to prove. Here's the hypothesis, and here's the data. So we had the poster session, and like 30 or 40 people presented posters. And then the next year, they said, hey, we'd like you to come and give a talk, a 30-minute talk. So I said, ooh, this will be good because, there's like 150 leaders in the company are here. The vice president, the big vice president of analytical holds this. I said, this will be a great chance for me to talk to them because, a lot of people knew me, but they hadn't really seen me talk about something. So I put together, I get 30 minutes to talk. We're in Cambridge at Robinson College. So, one of the newer colleges there. I had the second day. I thought it was a great spot. It was the first talk after lunch, the second day. So the lights go out. They said, hey, I get up there. They're saying, here's the podium. When you're 2 minutes out, this red light is going to come on. It means you better be on your last slide or ready to wrap up. So it's pitch dark. I'm going through it. I'm on my last slide. This red light comes on, and it looked like the freaking sun to me. It was just like a little tiny thing. And I'm like, oh my God. I finish up, and I said, boy, I hope everyone enjoyed it. They turned the lights on. First off, the hall's half empty because people are not back from lunch or they're planning on leaving. The other half that was there, half of those guys were asleep because we always have a big dinner and visit to a pub the night before.
Right.
And the few people that were awake said, "Hey, you did great, Ed. I loved it." So, so, so the third time, so the third time I go over, they asked me to give the keynote speech after dinner
Hey, wow.
because
That's a big deal.
a lot of these guys got to know me better and said, "Hey, he's kind of got a wacky sense of humor." He'll from this, so I gave a talk called a Yank's Eye View of SmithKline Beecham and stuff. And I put together a bunch of funny slides and stuff. And just before we get ready to talk, the slide projector doesn't work. This is
Oh,
slides,
I
the old
know.
school slides, right?
Yeah,
This is
yeah.
a computer plugged in.
No,
These are
it's not a
slides. So
projector.
there's
Yeah.
something wrong. So I have to, they said, well, what can you, so I did like a 20-minute stream of conscious thing, entertaining these guys until the slide deck got up. And then I did my presentation and stuff. And then the last time I went, they said, you've done a poster, you did a talk, you did a keynote, you can just show up and enjoy yourself. And I did. And then we merged with Glaxo and they changed the structure. I never went to that again. But yeah, that's a key thing. If you're going to be a leader, you have to be comfortable talking.
Yeah,
You have to be able
and
to be very comfortable. You need to be able to talk about good stuff and bad stuff.
yeah,
And that's a key thing. And I knew of another guy. He was in a leadership role. He virtually had panic attacks if he had to talk. And
wow,
you could tell that it was not someplace he got energy from. It was very trying, and it was like, this is kind of going to be a block for you.
yeah,
That you're never going to get any higher because the expectation is you can get up and talk about whatever they need to with very little notice. He had to do a lot of planning if he was going to do a presentation. Where I said, let me read the slides once, and I'll go.
right. Well, and you just mentioned something too about having hard conversations. It's like to be able to either speak in front of a large audience or have hard conversations with one person. I mean, sometimes
Yeah,
that can be just as debilitating for some leaders is not being able to say things that you got to say that sometimes is not great news or
yeah. It's
doesn't
always
have
a
good
tough thing,
outcomes.
but like I said, the benefits of being in the leadership spot way outweighed the ones not, because I got to
Sure.
do stuff that I wanted to do and
Yeah.
how I wanted to do it, and often it worked out pretty well. So I was able to get these guys to pay me for 41 years. So that's got to be worth something.
Yeah. You brought value to the table, I think.
Yeah, and the other part, I went back. One of the ways we used to introduce ourselves, we had a one slider called a career journey, and basically on the top it maps out kind of like how you got to where you are now, and the bottom is like, well, not just how did you get there, how did you really do it? What motivates you, what gives you energy, what are you really proud about doing? One of the things that when I was younger, I used to get a lot of work assigned to me because I always delivered.
Right.
In fact, one of the VPs used to call me FedEx Ed because I always delivered. He would ask me for stuff, and I would get it done and stuff. That's not a bad nickname, right?
That's a good
That's
one.
not too bad
FedEx Ed. Yeah, I like
because
that.
I would deliver, and
Yeah.
that's a key thing. And the other part
Yeah.
is, can people trust you?
Yeah.
We have a lot of relationships here. A lot of stuff has to be based on trust. I trust
Yeah.
you to do this.
Right.
In fact, I always tell people, I'm going to ask you to do something. I will likely not check up on you because you said you're going to do it. But if you don't do it, I will likely never ask you again
Right.
and stop, because I'm not a micromanager. I work with people that used to say, let's discuss this down to the third decimal place. And I'm like, oh my God, we've got all these smart people. That's not the direction and help they need
No.
to do that. It's very demotivating. So
Yeah.
I like to deliver stuff saying, it's kind of the West Point thing. They talk about all these strategies, but the general says, I want you to take this hill. He's not going to say, march 2 miles here and 3 miles here, dig a trench here, do this. He says, I want you to take that hill. And then we figure out how to do that. I want you to figure out the control plan for this drug. Well, we know the regulations. We know how the drug was developed. We've worked closely with analytical sciences for method development. I could write a control plan for a typical monoclonal in like 3 minutes now because we've done it so much. Probably AI could do it faster.
It's 3 seconds. 3 minutes to 3 seconds now. Yeah.
Exactly.
Yeah. Oh, so interesting. Like it's you bring great perspective to your from your journey, and I'm really grateful for you to share it with me and listeners. And I think you had some really great advice to pass forward, and I'm sure there'll be some appreciative people out there hearing
Yeah.
your story. So,
So it's tough. I mean, it's always been tough in the science world because
hmm
if you're in the for-profit stuff, they will think of nothing of shutting you down and moving. A key asset fails,
yeah,
you're out. That's the advice I always give people: stay current. Don't be stuck in something that worked even 2 years ago. You need to constantly stay up to date, not only the technology but the regulatory space. That shapes so much what we do. There's
right,
so many new things. I mean, I'm still learning about how the CAR T stuff works. Right now, we have clients that are doing similar stuff we've done. They want advice on how does this look to you? Read our BLA and give us some feedback. Are there gaps do you see? Or if you don't see gaps, what could be better? How do we defend this practice to when we get an inspection done,
right.
or this is kind of weak. What would we need to beef this up? Those are kind of the stuff that I'm working on now. And it's like I said, it's pretty cool. You get to do all kinds of stuff. If you don't want to work, and so tomorrow my family and I are going to Disneyland.
Nice.
It's just a long plan. We have annual passes, so we go whenever we can. We couldn't go a couple weeks ago, so we're going tomorrow. So I'm not going to work.
Good for
And
you.
I've told the people, hey, don't bother me because I'll be hopefully on Big Thunder Mountain or eating a Dole Whip or something.
Well, you enjoy yourself, and thank you again for coming on this episode and sharing a little slice of your world with us. I really appreciate it.
Yeah, I can't wait. I can't wait to see what the final product looks like, and it's really interesting, Heath. I was really excited to do this because who doesn't love to talk about themselves?
Especially when you went to acting school, you have to
Well,
be able
that was
to
high school.
show.
I wasn't very good. I think the role I had was I was a townsman and the music man. So I was an Iowa townsman
Is that
and
right?
stuff. But like I said,
And it
if
was
you're
a
going to do,
musical,
yeah, but
so you
you're
can
with
sing and
dozens of
dance
people.
too. Like that.
No, there was no dancing. It was pointing and trouble.
Trouble in River City.
But yeah, it's really good. To be able to speak easily in public.
Yeah, it's
Is
definitely yes.
really a real differentiator because I work with people, like I said, were really smart and they were tortured if they had to get up and talk.
And it's learnable. It's not necessarily. It's not innate in everybody. It's something
Yeah,
you can learn and something you can practice and get better at. So
yeah.
yeah, it's great advice. It's great advice. All right, Ed. Thanks very much, and we'll talk again soon.
Okay.
All
Thanks a
right.
lot.
I really appreciate Ed. His storytelling capability is obvious. I really liked what he said about leadership style, and that's staying authentic. He is who he is, and he has a very clear idea of when there's a goal to be reached that everyone can do their job, and when you say you're going to do something, you do it, and I think that's a real great takeaway for a leader. Sometimes leaders have a little bit too much micromanagement going on, and I think that's good advice. He also, reaffirmed that why most people in the health tech space, why they do what they do is because they're looking for some sort of human outcome for somebody else, a way to improve someone else's life. And sharing that story of the woman about her son getting the prescription drugs that helped more or less save his life. A piece of advice I've never heard before from somebody in upper management is acting lessons and learning how to for public speaking purposes. I mean, that's a great piece of advice. If somebody is struggling with doing public presentations or public speaking or giving talks or lectures or something like that, or even a podcast, for example, taking an acting lesson. You don't have to go to acting school, but I think a little bit of acting and learning how how to present is great advice. So I think that's super valuable. Anyway, I really enjoyed meeting Ed, and I hope you did too. And thanks for listening, and stay healthy.